Chapter 18, Section 3, Pathways of Amino Acid Degradation. Now, amino acids, hence the proteins you take up in your diet, you're looking at about 10 to 15% of the energy that you produce comes from the amino acids. As we talked about in previous chapters, a lot of it's going to come from the fatty acids, the lipids. A lot of quick energy is going to be coming from the carbohydrates. And depending upon your diet, those levels can change depending upon, are you doing the American diet? Are you on the keto diet? Are you on a vegetarian diet? And how you're looking at and, balancing all those different things. What we do know is when it comes to the amino acid catabolism, you're looking at about 20 different catabolic pathways, all converging. Because think about it, the past, 5 chapters, and we've talked all these different pathways and stuff. 6 words, 6 names keep coming up. Pyruvate, acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate. I've been trying to hammer it home. None of this stuff, you may have learned it in intracellular molecular, cell biology, whatever. You may have learned individual pathways, but these pathways are not individual. They are not independent of each other. You may start off with the molecules that are going through the citric acid cycle, but because of changes in the environment of intracellular environment of the cell, changes at the organismal level, those same carbons that that were going through the citric acid cycle may be shunted over to gluconeogenesis, or something that was going through gluconeogenesis may be shunted over to fatty acid catabolism, or fatty acid buildup, or amino acid catabolism, and so on. So things are constantly moving. It's not just here, we're in this and we're staying in this. No, we're here, now we're over there, now we're back, now we're in something different. And yeah, it is very, very hard to track, which is why we teach everything independent. Here's this cycle, here's this cycle, here's this cycle. Knowing in our minds that this cycle overlaps with this one, and those two overlap with this third, and then there's a fourth one that may feed in every so often, but only under certain conditions and certain phases of the moon. I mean, look here. Basing everything around the citric acid cycle, okay, because this is one we know. We're second to glycolysis is the one we're most familiar with. All of the amino acids in a ketogenic or a glucogenic means can feed in at some point. Pyruvate, oxaloacetate, alpha-ketoglutarate, succinyl-CoA, acetyl-CoA. This is just the amino acids. You add in, the fatty acids, you add in the carbohydrates, and yeah, this becomes very, very messy. And keep in mind, these arrows are also, for the most part, reversible. Succinyl-CoA, with a twist to tweak, not shake of the head, becomes any of these amino acids. Alpha-ketoglutarate, with a twist to tweak and a shake of the head, becomes glutamate, and the glutamate can become any of these. Pyruvate, which can become oxaloacetate very easily, well, that pyruvate can, twist to tweak, shake and nod of the head, becomes any of these, and so on and so forth. Notice they were color-coded, ketogenic and glucogenic. The ketogenic means they're going to yield ketone bodies. We talked ketone bodies last chapter at the end of chapter 17, the acetone, acetoacetate, those things like that. So the glucogenic, these are ones that can be readily converted into carbohydrates by the liver. Because remember, in us, only our liver does this. So you can take, all the amino acids except for lysine and leucine, twist, a tweak, cut, a paste, nod of the head, and gluconeogenesis. Off that goes. Cofactors that we have talked about many, many times. Biotin, okay, remember this one, transfers carbon dioxide, holds carbon dioxide so it can be added. So your 5-carbon skeleton could become a 6-carbon skeleton. Tetrahydrofolate, right here, folate. You'll see this sometimes just referred to as folate. Transfers intermediate oxidation states. S-adenosyl blah 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 methionine transfers methyl groups. We also have the biotin and so on. So the biotin I just talked here, folate, and behind my big old not quite bald head right now, what they refer to as adomet, S-adenosyl methionine. It's an adenosine nucleotide with a long carbon chain in replace of the phosphate group. The tetrahydrofolate, you've got pterin, which is 2 carbon rings. I'm sorry, 2 nitrogen rings. Carbon rings. I forget how you would phrase this one. Do you call these carbon rings or nitrogen rings? Carbon bases. Anyways, you have 2 6-sided rings that attach to each other. This is synthesized in bacteria more often than not. You'll have a version of this. It is going to be in folate, which is made or further processed by plants. Some plants make it themselves. A lot of them take it from the bacteria in the soil that they're associated with. We, animals, get it by eating plants. Or if you're really good, you eat an animal that ate a plant, for those of you who are truly carnivores. Okay, when it comes to tetrahydrofolate, the folate, depending upon which amino acid it's going to be interacting with, well, different carbons will be bonded. You can see here as it's saying, N5, N10, or both, depending upon, well, you're looking here at serine, glycine, you'll see here N5, N10, or if you're going, formate here at 10, which will then be converted, you get most highest oxidation levels, lowest oxidation, too high, too low, just right, I don't know. I'm going to admit, my organic game is a bit weak when it comes to this one. So as far as I'm concerned, what you need to understand is that the tetrahydrofolate, the folate, it's acting as, holding the intermediates, and it will, where it will hold it, where the interaction will occur, will be different depending upon which substrate, which enzyme. Either it's the 5, the 10, or both. There. That's the level I want you to know it at. The adomet methyl groups is what you're looking at here. Methionine adenosyl transferase catalyzes the synthesis of our S-adenylmethionine from ATP and methionine, which makes sense. Adenosyl is AMP taking from ATP that's had the terminal 2-phosphate removed from it. S-adenosyl homocysteine, when you take the S-adenosyl this and you transfer it to an acceptor, this is what's left. When you go through and you look here, S-adenyl homocysteine. Hydrolase, remove the adenosine, you get homocysteine. Homocysteine interacting with our folate gives you methionine. Methionine, ATP, gives you S-adenyl methionine here. Transfer of some methyl from one to another. Take this methyl, transfer it to this target one. Boom. In the process, our strand here, 1, 2, 3, 4 carbon, sulfur, then our adenosyl, 1, 2, 3, 4 sulfur, adenosyl. What's missing is that methyl group right there. We don't even call it part of the carbon chain. We're just calling it a methyl functional group because it's not directly attached to the carbon chain. It's now a functional group. So that functional group is transferred to some target. Okay, so S-adenosyl methionine becomes S-adenyl homocysteine. Homocysteine will be released from the adenosine nucleotide. The homocysteine will be remethylated because folate has taken it, and methyl tetrahydrobutyrate will take it and then transfer it over. We get our methionine again, interaction with ATP, S-adenosyl methionine, and we do it all over again. Your textbook likes to bring up different human diseases, human deficiencies, based on a lot of these metabolic pathways. Well, one of them, as we see here, is, deficiency in B12, the folate. Pernicious anemia. So what happens is you get the cysteine that was just cut off of the S-adenyl cysteine. Remember, that gets the nucleotide gets cut off, leaving you just cysteine, and then it's supposed to do the rebuilding, where the cysteine interacts with, the folate, gets another methyl group, and becomes methionine. The methionine, which will then go and, interact with ATP, give you the S-adenosyl methionine all over again. Well, they don't have that part that allows for the B12 interaction to go from cysteine to methionine. So they get a buildup of cysteine and a lack of methionine. So this whole taking the methyl group and adding it doesn't happen. Other things you can have, megaloblastic anemia, because of the deficiency in the B12. Either you can't take it up or it's completely missing from your diet or something. You're going to have your erythrocytes, your red blood cells, are not going to mature. So, you're going to have production of the precursor cells, but no mature ones. So you're going to have all these immature megaloblasts in the bloodstream, none of them maturing, nothing making it out into your bloodstream, hence the anemia. So you're going to get a large number of mature ones, but the overall large number of oversized ones. But if you were just count the true number of red blood cells, big, small, whatever, the population is depressed, is lower than it should be. And you're getting just this increased number of huge ones instead of numerous, normal-sized ones. Turns out these macrocytes have issues when it comes to carrying oxygen. maintaining oxygenation levels. So it's almost as if you just have a decreased population number overall, even though you've got these bigger ones. Turns out having a bigger cell doesn't compensate for not having the normal cell number. The Tetrahydrobioparent. Similar to Biotin you see in the Tetrahydrofolate, but this derivative of the folic acid is going to be utilized in oxidation reactions of amino acids. So our 6 amino acids that are degraded to pyruvate: alanine, tryptophan, cysteine, serine, glycine, and threonine. Okay, they're converted in whole or in part to pyruvate. Certain ones of these are only 3 carbons in length. Some of them are more, so there's going to have to be some cleavage here and there. The pyruvate either then becomes acetyl-CoA, goes into the citric acid cycle, or these are going to be large enough or almost large enough where you can just add either a carbon dioxide or a carbonate onto it, and then you can get oxaloacetate, which can then go to gluconeogenesis. And here's the different, tryptophan through numerous steps can become, here you get this cleavage of these chains off here, and you get alanine. Alanine can become pyruvate. Just got to pull off that amino group. We've talked about this last section. Or you can take cysteine here, pull off the amino, pull off the sulfur thiol functional group. And then you have pyruvate. Pyruvate, serine to here, and so on. Pyruvate can become serine, serine can become glycine, boom, 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 or this way. So the pyruvate seems to be the interaction. There are some, I can't remember the steps, I think I remember reading where, serine can be converted to alanine. You just have to go through a pyruvate intermediate. Serine to pyruvate. This is reversible, that's reversible. Not by the enzymes stated here. There are other enzymes that will take this and do that. The serine dehydratase catalyzes the conversion of serine to pyruvate. All right, so what do you got to do? Well, you got to take the serine, and you're going to go through multiple steps. Remove the beta hydroxyl and the alpha amino groups. We've got, hydroxyl, and you have an amino that have to be removed stepwise. And I'm not going to go through the different steps here. You know by now the overall process that this could be looking at. Glycine is going to go through many of the same steps, or its versions of.